Okay friends, so now we are going to start another lecture on microbial virulence factors, exotoxin and endotoxin. In the last lecture we talked about some of the highlights of the virulence factors, right, which are leading to the pathogenicity. Here we will discuss some detail. So we will talk about the microbial virulence factors, toxin, endotoxins and exotoxin. The first lecture will be on microbial virulence factors. Now, the microbial virulence factor means the factors responsible for pathogenicity are infection causing properties, attributes which are causing the infection in the host after the microbes attack. So, by definition, virulence factors are cellular structures, specialized strategies, or molecules produced by the pathogens, right? Strategies, specialized strategies means how they adhere and how they penetrate. What are the strategic approaches they adopted by pathogens? So we call the specialized strategies produced by the pathogens, and they enable or they facilitate to colonize the host and evade or suppress the immune system or immune responses. It acquires the nutrients, adapt to environmental changes and cause the diseases. Now, the microbial virulence factors are basically the factors which promote the infection by the microorganisms in a particular environment, and they also adapt the changes in the environment, and they also acquire certain specific conditions through plasmids, through harboring some genes and so on and so forth. Now, they're essential for infection establishment and survival, persistence and transmission, because they are essential for the infection establishment. Means they have full factors are responsible for establishment of infection. Means like infection which is completely established and it causes damage to the host, and moreover host may not defend to that infective microorganisms. So this is called establishment. If the host start defending, then establishment will not be called as establishment or established infection. They are transient infection. Now survival, it survives for a long time, it persists for a long time, and then it also transmits from one host to another host. So this is the whole cycle of infection spread. The general characteristics of the virulence factors are genotypic diversity, high mutation rate, so that it may not be catched by the defense system. Horizontal gene transfer, so they can transfer from not vertical but horizontally, they can transfer through plasmid, through extra chromosomal material from one bacteria to another bacteria, even across the genera or across the species. So they also introduce the variation supporting adaptation to host defense, and this process of variation and gene acquiring variations through plasmids, through TN elements, through IS elements, they provide the adaptation to the host defense system. So the host defense system, and this is how we call them as established infection. Then phenotypic heterogeneity. The phenotypic heterogeneity is antigenic and phase variation allow the pathogens to escape, right? And immune recognition, right? So pathogenic, the phenotypic heterogeneity means like antigenic and phase variation allow the pathogens to escape immune system. So basically it is all like giving a dodge to the immune system, because when the bacteria enter into the cell, the immune system get activated, and when the immune systems get activated, then this immune system is basically responsible to combat with this kind of a microorganism, incoming microorganism. So what it does, it keeps changing the antigenic phase variation in the antigenic property, and allow the pathogens to escape the immune system of the host. Immune system of the host could not recognize such kind of a variation, such kind of a shift and drift. The molecular arsenal, it's a wide range of protein, enzymes and toxins acts individually or synergistically to enhance the pathogenic success. So wide range of proteins, enzymes means like produced by the bacteria, and the toxin produced by the bacteria, right? And they individually are synergistically means like with other bacteria they are living with to enhance the pathogenic property. For example, sometime one bacteria is promoting the pathogenicity of the other bacteria. So this is called a synergism between the bacterial properties to cause infection or to cause high pathogenicity. Now, the dormancy and persistence. Dormancy means like sometime pathogens go to long latent stage. We call formation of persister cells or biofilm support, where the large number of cells can remain in the protected manner. And chronic infection may cause by such kind of a dormancy because it's a long time infection, long-term infections and antibiotic tolerance. Now, the functions of virulence factors, they facilitate the colonization of a host tissue. This is one thing. It also evade or suppress the immunity. So the microorganisms can escape out from this kind of a system, and it suppresses the immunity, both innate and adaptive immunity, cell-mediated immunity and innate immunity and adaptive immunity. It acquires the nutrient, especially iron, to grow, to live, and to sustain. It promotes the invasion and dissemination without loss of tissue. So this is also very important. Without losing the tissue, host tissue, it can it promote the invasions and dissemination out from it. So we call them exocytosed, endocytosed, and exocytosed. Both the process can be taken by this and cause tissue damage and pathogenicity. It also causes the tissue damage and pathogenicity. It supports the persistence and chronic infection. The persistence and chronic infection through dormancy or biofilm. So it can persist for a long time, right? And they can also support the chronic infection. Chronic infections, as I talked in my last lecture, that chronic infection means the infection which is for long time based infections. And acute infection is the quick infections and early stage infections. The major categories of the virulence factors. What are the major categories? The adherence, colonization factors. Pathogen must attach to the host surfaces and to initiate the infection, right? And what are these? Fimbriae, pili, which mediate the binding process, and E. coli type 1 fimbriae, type 2 fimbriae, and many other type of fimbriae are present in different bacteria. Then afimbrial addition. Afimbrial addition means like bind the extracellular matrix protein, which is on outer side. So we call fibronectin, and we call laminin. So these are basically helps to dissolve certain tissues and to get enter it into that. The biofilm formation is again important. The collective growth mode providing the resistance. For example, the Pseudomonas aeruginosa and in gram-positive, we have several bacteria like Staphylococcus aureus and as mutans. Specialized systems, OPA proteins, Neisseria gonorrhea, they are specific signal for virulence factors or for pathogenicity of the Neisseria gonorrhea strain. Then filamentous hemagglutinin like Bordetella pertussis in pertussis fever. The hemagglutinin is basically the protein factors which are filamentous protein factors which is called as one of the virulent factor in Bordetella. Capsule is again virulent factor. Capsule is a surface component. So capsules and structures protein, they protect bacteria against the host defense system. So capsule is like a outer covering of the cellular covering of the bacteria, and which cause the protection from the bacteria from other host system, and they can evade the host defense systems and can survive and can cause the damage to the host system. Now, polysaccharide capsules, they inhibit the pathogen phagocytosis, S. pneumonia, Klebsiella pneumonia, Staphylococcus aureus, and Streptococcus pneumonia. So these are the bacteria which are polysaccharide capsules and inhibit the phagocytosis. M protein is one where we call S. pyogen is example. It prevents the opsonization, right? So it prevents the promotion of the catenation, promotion of these bacteria to grow in that environment. Protein A is basically S. aureus example. It binds the Fc region of IgG. So this is very important. So when it binds to IgG, it will be again protected from the immune system of the host. Then mycolic acid in case of Mycobacterium, it resists the lysosomal degradation. So because in host system there are lysosomes present which produce the enzymes, lysosomal enzymes which degrade these bacteria. Sometimes they engulf these bacteria and dissolve them in the vacuoles. Sometimes they release enzymes and degrade the bacteria. So mycolic acid is basically protected from these lysosomes in the Microbacterium tuberculosis or Microbacterium leprae. Now, invasion and evasion factors. What are the invasion and evasion factors? It facilitates the host penetration and immune evasion. So it facilitates the host penetration means like host cannot be protected themselves because it increase the process of invasion. So the penetration process cannot be controlled by the host factors. So bacteria has this invasion property which facilitate or which enhance the penetration process, and also it immune system is escaped from the immune system of the host. So immunomodulation will not work here, and bacteria can survive even having that immune system. What are the invasion enzymes? Hyaluronic acid, which is present in the cementing material in the collagens, another cementing material is present in the intercellular space, dissolved by the invasion enzymes like hyaluronidase and collagenase. DNase is another one, streptokinase and neuraminidase. So these are the enzymes which helps to microorganisms in viruses and bacteria to enter into the cell membrane. Like salicylic acid is dissolved by the neuraminidase. Streptokinase is another enzyme which promote the virulence factors in the bacteria to cause infection at the site of wounds. And DNase is dissolve the DNA, extracellular DNA, degraded DNA, present in the between the two cells. So it produce the viscosity, increased viscosity, and this viscosity can be reduced by producing the DNase, by dissolving the DNA, extracellular DNA. Now, intercellular survival mechanism is something listeria. Listeriolysin O escape the phagosome. So, the listeria cannot undergo in the phagosome formations, or phagosome cannot engulf these listeria by producing the listeriolysin O, which can allow to escape from the phagosome, and listeria can survive and cause the damage to the host system. Salmonella is another example. Salmonella typhimurium is the species where modify the vacuole for the survival. So they can modify the vacuole so that it may not damage the host cells are deceived by this salmonella, and salmonella can survive in the host cells or host system. Microbacterium is block the phagosomes, lysosomes fusions, and so they can protect it in the host system. Toxins and exotoxins they are the neurotoxins, for example, Clostridium botulinum. Then enterotoxin, we have Vibrio cholera. Then we have cytotoxin like Clostridium diphtheria, and endotoxins like gram-negative LPS trigger the systemic effects. So the two categories of these toxins like exotoxins and endotoxins are produced by the bacteria to defend and to cause more severe infection into the host cell. The antigenic variation, trypanosoma VSG switching, Neisseria like Pili variations, and influenza viruses like HA and NA drift and shift. So there's a shift, genetic shift and drift takes place in hemagglutinin and neuraminidase in case of influenza virus, which is very common, and they protect the viruses from various drugs, from various enemies by having variation in the antigenic in the HA and NA functions. The other virulence factors like iron acquisitions like cytophores, example in enterobactin, quorum sensing mechanism controlled by several genes present in bacteria, regulate the virulence genes and expression. Pseudomonas is one of the examples, and also secretion system is one of them, where the T3SS, Salmonella, Shigella T6SS interbacterial competitions and virulence takes place by secretion system. So, iron acquisitions, column sensing and secretion systems are also very important. Now, the toxin antitoxin system, the plasmid stabilization, stress survival. So, stability of plasmid means like if plasmids are stabilized and they can have the intact gene which are causing the support to the bacteria, and they also transferred from one bacteria to another bacteria. We call horizontal gene transfer, and they remain stable with the gene intact with the plasmid. Let's talk about the toxins. These are the poisonous substances that some microorganisms they do produce and contribute toward the pathogenicity. Toxigenicity is the capacity of microorganisms to produce toxin, and toxemia, the presence of toxin in the blood. What are the effect of toxins? It produces the fever, allergic reactions, cardiovascular disturbance, diarrhea, shock. It inhibits the protein synthesis, destroy blood cells, blood vessels, and disturb the nervous system by causing the spasm. So all these, the outcome of these toxins, which are basically responsible for the damaging to the host cell. Antitoxins, in the toxin you have two type of toxin, endotoxin and exotoxins. The endotoxins generally gram-negative bacterial toxins, where the endotoxins are lipids, part of the outer membrane, gram-negative bacteria, and components are lipopolysaccharide. And toxin proportion is a lipid A protein, or lipid A component. It released during the bacterial lysis. So cell death is caused by the endotoxin. Now, during the bacterial multiplication, they also release, right? And unlike exotoxin, which are endotoxins are not secreted protein, but a structural component, right? So this is how the endotoxin example here, lipid portions of the lipopolysaccharides that are part of outer membrane and cell wall of gram-negative bacteria, lipid we call lipid A. The endotoxins are liberated or released when the bacteria die and the cell wall lyses. So they are present on the outer membrane of this and they break apart. For example, salmonellas, typhimuriums, an example of a gram-negative bacteria that produce the endotoxins, and these endotoxins they composed of a lipid that are part of the cell wall. Now, endotoxins and pyrogenic response. How does the endotoxin cause the pyrogenic response? Pyrogenic response means like a fever. Pyrogenicity means increase in temperature. So basically bacterium engulfed by these macrophages, and then endotoxins are released, right? Once the endotoxins are released, the cytokines are produced in blood vessels, and these cytokines induce the hypothalamus of brain, and which is called the prostaglandin of the hypothalamus, and this prostaglandin then cause the pituitary gland give the action to the prostaglandin, and this prostaglandin is basically cause the fever. So the process of this pyrogenic bacteria, the cause of toxin effect is basically when the macrophage ingests these bacteria, and the bacterium degrades the vacuoles, and after degrading the vacuole they release some endotoxins that induce the macrophage to produce the cytokines, and the cytokines like interleukin, IL-1, then tumor necrosis factors, and then TNF alpha, gamma, and these cytokines are released in the blood streams by the macrophages, and through which they travel to hypothalamus, and then the temperature control center for brain, and then the cytokine induce the hypothalamus to produce the prostaglandin, and this prostaglandin which rests the body's thermostat and to the higher temperature producing the fever. So this is the whole cycle and process of how the fever takes place, how the pyrogen effect takes place by the endotoxin. Now, mechanism of action of these toxins, they stimulate the macrophages, the axis of cytokine release, right, and like TNF-alpha, IL-1, gamma, cytokine storm we call toxic to host. The main effects are fever, like pyrogenic responses via prostaglandins, and shocks, anaphylactic shocks, septic shock, and due to the TNF alpha, gamma, and induce which induce the vascular permeability, which reduce the blood pressures, and so person can go for the shock. The dissemination are disseminated intravascular intravascular coagulations, like we call DIC. Dissemination of intravascular coagulation, like small clot formation takes place, and then they form the tissue death. And so if it is increased, they may cause major damage to the host cell or host system. The blood-brain barriers weakening, so CNS central nervous system infections more likely caused by these blood-brain barrier breaking are weakening. So when the blood-brain barrier will block the toxins, they can reduce the capacity to get entered these toxins, and so it allows the release of this toxin to the brain, and the brain get infected. The miscarriage can be triggered. So miscarriage is during the early pregnancy or mid-pregnancy, the miscarriage takes place through these toxins. The clinical manifestations of these toxins are the chill, fevers, and muscles ache, weakness, in shock, like a rapid fall in BP, and then organ damage, kidney, lungs, GIT, gastrointestinal tract. In septic shock, high mortality, about 30 to 50%. Same general symptoms, like regardless of bacterial species. Then representative organisms like Salmonella typhi, typhoid fevers, Proteus species, and then which may lead to the UTI infection, and then Neisseria meningitidis. This is called meningococcal meningitis. So these are all deadly diseases or deadly infections caused by these bacteria, and they are emergency infections. Other gram-negative bacteria or gram-negative pathogens are E. coli, and then Haemophilus influenza, and many more, which are cause. Now, representative organisms are Salmonella typhimurium, causing the typhoid fevers, then Proteus species causing UTI infections, Neisseria meningitidis is meningococcal meningitis. Other gram-negative bacteria are gram-negative pathogens like E. coli, Haemophilus influenza, and so on. The immune responses are endotoxins that do not induce the strong antitoxin. Antibodies forms but are usually ineffective or may enhance the toxicity. Its recovery depends on host clearance, example liver, lipase breakdowns, and endotoxins. So this is the immune response against that endotoxins. The laboratory detection. How you detect in the laboratory? Now the lamellus amoebocytes lysate. This is called lamellus amoebocytes lysate. We call LAL assay. This is a simple laboratory assay where we can detect the kind of infection in the blood, in the urine, and other. So use blood of a horseshoe crab, Lamellus polyphemus, and this is amoebocytes lies is in presence of endotoxin like clot and formation. So this is the test where the endotoxin can show the expression of its effect through these setting, through these things like amoebocytes lice, and then a horseshoe crab in case of Lamellus polyphemus. Highly sensitive because it detects the minute endotoxins. So in a very minute endotoxins like botulinum toxins, tetanus toxins, they're very minute amount. Toxins can contamination in drug, fluids, medical devices can easily be detected. Now, so in this topic, what we discuss about the various virulent factors and how these virulent factors help the host to invade or to escape from the pathogen. So one of the most important point here is that how these hosts can be can lose their ability to defend, or how lost their ability to control the infection or infective microorganisms to get enter. So the whole thing is based on the penetration and evasion from the host mechanism. Once it penetrates, well, if it is engulfed by the host system, or if it is host system get overpowered and defend this kind of a cellular system or bacterial system or virus system, then host will not be damaged. So in order to fully damage the host, it is important that to completely lose it. Okay, with this I stop here, and thank you very much. We will start another lecture soon.